this year. We're interested in single molecule fluorescence microscopy. So today I'm going to be talking to you guys about a new microscopy technique, convex lens induced confinement, and its application to probing DNA confinement physics. The confinement physics of DNA is a fascinating subject. If we consider the nucleus of a cell as about 2 meters of DNA crammed into a 6-micron diameter nucleus. That's not even accounting for all the other proteins that are present. In fact, some of these proteins, such as this one on the right, even help DNA take on more compact configurations. Understanding the physical properties of individual DNA molecules is an important biophysics question, as it forms a wide range of physiological processes, such as the kind of conformations that DNA acquires affect its reactions with transcription factors or repair proteins. To help better understand this complex physiological system, I performed in vitro experiments on a very simple model. I used fluorescently labeled DNA. To perform my experiments, there are 2 key things that I need to achieve. Number 1, I need to be able to resolve individual DNA molecules so that I can watch their fluctuations and conformations in time. And number 2, I need to be able to subject these molecules to a wide variety of confinement, ranging from the nanometer to the micrometer scale. One existing approach to this is the use of these nanochannels, or nanoholes. Via the use of electron beam lithography, you're able to produce these features in fused silica substrates as small as 20 nanometers in cross-section. There are, however, some limitations to this approach. Number 1, the production of these chips is very expensive. Nanofabrication facilities are very hard to come by. Also, the production of these lithography procedures is very time-consuming, especially at an undergraduate or graduate level. Finally, these chips that are produced are of discrete dimension. In probing DNA confinement physics, it would be ideal to be able to visualize a continuum of different regimes, so we can understand how DNA behaves in these transition periods. To overcome this, my lab uses a new technique called convex lens induced confinement. It's based on a very simple principle. We confine molecules to a thin film. This has the advantage of suppressing background fluorescence and extending observation times. How we do this, it also provides a great platform to visualize DNA under confinement. How we do this is we use a lens here that you see mounted on a piezo actuator. This lens is then brought down into contact with the top cover slip. This top cover slip is then flexed until it comes into contact with the bottom cover slip. At the point of contact, there's about 2 nanometer separation, as set by the surface reference. From there on, there's then a gradual increase in the height from this 2 nanometers to many, many microns. So this provides a platform in which we can look at DNA under a variety of different confinement regimes. If we were to look at the chamber uncompressed and we were to visualize the fluorescence profile of the DNA, you can see that we're unable to resolve individual DNA molecules. This is due to the out-of-focus contributions of many, many layers far beyond the focal depth of the objective. When the chamber's then been formed, if we look at the fluorescence profile near the center, we can see that we can easily view individual DNA molecules. We can watch their fluctuations in time and understand how they behave. In studying DNA as a function of confinement, it's really important that we know how much confinement they're being subjected to. To do this, we perform interferometry of the chamber so that we can precisely measure the chamber height everywhere. Here, you'll see a video of interferometry as the chamber is being formed and the top surface is flexed down to touch the bottom surface. What you'll observe is the formation of Newton's rings. This is due to reflections off the top and bottom surface that create constructive and deconstructive minima and maxima. You'll see at the center a destructive minima, and just outside of that, the first constructive maximum. By knowing the index of refraction of the media and the wavelength of the light source used, we can then determine the position and the height of each one of these dark minima. In addition to that, we image the fluorescence profile of a very small, freely diffusing dye. This dye is characterized by a diameter of about 1 nanometer, so it's able to penetrate to very small chamber heights. And as such, its fluorescence profile is proportional to changes in the chamber height. Here you'll see a video of the fluorescence profile as the chamber is formed. By fitting an ellipse to this fluorescence profile and using the location of these dark minima, we're then able to extract a continuous map of the chamber height everywhere. And as such, we're able to understand the amount of confinement that we're applying to the DNA. So now that you understand the device and the techniques, let me take you through an experiment. We start with the chamber originally uncompressed, and we load our sample. We then slowly flex the top surface down until it's in contact with the bottom surface. Up on the top right, you'll see a schematic depicting the chamber geometry. You can imagine that at small heights, the DNA becomes much more stretched out as it deflects up and off the bottom surface. And at larger heights, the DNA takes on these more sort of bundle topologies. We then perform a three-step experiment. Number one, we image the fluorescence profile of the DNA at a grid of locations. That's shown on the far right. We then image the fluorescence profile of a spectrally separate dye in the same chamber, shown in the middle. And finally, we perform the interferometry of the chamber. As I mentioned before, using the dye and the interferometry, we can then extract a map of the chamber height everywhere. So let's now turn our attention to the fluorescence profile of the DNA. You'll notice that below that green ring, the DNA concentrations become suppressed. Well, the diameter of gyration, the diameter of the ball of the DNA, is about 1 micron. And that green ring corresponds to a micron in height. Below that, there's a reduced number of configurations accessible to DNA, and hence an entropic penalty associated with accessing those regimes. If we were to look at DNA at different heights in the chamber, such as the bottom left picture there, taken around a vertical height of 100 nanometers, we'll see that DNA has become more stretched out, as we expected. It's deflecting off the surfaces. If we were then to compare that to one taken out closer to 700, we'll see that the DNA becomes more bundled up. So we've seen that below its diameter of gyration, the concentration of the DNA becomes suppressed at the center of the chamber. Because the concentration has been suppressed, so has its intensity. And we can actually use the intensity profile of the DNA to extract the free energy of confinement from this. So here, you see plotted the intensity profiles of the dye and the DNA as a function of chamber height. In red, the dye intensity is linear with the chamber height. However, in blue, the DNA intensity is only linear down to about a micron. As I said, this is the diameter of gyration of lambda DNA. Below there, it has to take on an entropic penalty in order to access those lower regimes. We can then relate the intensity to the concentration of this top equation. And using the bottom one, we know that the concentration is proportional to Boltzmann's free energy of confinement. With some simple rearrangement, we can arrive at this bottom formula here, and we can actually plot the free energy as a function of chamber height. You see that, as you would expect, at small chamber heights, there is a high free energy of confinement. And as the chamber height approaches the diameter of gyration of the DNA, the confinement potential goes to 0. What's really interesting now is to consider the polymer physics of the DNA, to consider the different scaling regimes and the transitions with chamber height and the conformations that the DNA acquires. To do this, we take a log plot of the free energy that you see superimposed here. You can see that these different slopes correspond to these different scaling regimes. The first one, that's below the diameter of gyration, and the second one, that's around 50 nanometers. What's really interesting about the 50 nanometer point is that's the persistent length, the DNA that we were using. Persistent length of a polymer is the length over which it can be considered to be a rigid rod. So we see that as we approach this persistent length, there's a qualitative change in the free energy of confinement. Imagine that the DNA is only able to bounce up and off the top surface. It's not able to form coils in the one dimension. So, what did we learn from these experiments? Well, we've learned that we can use a simple platform, convex lens-induced confinement, to study systems under confinement, ranging from the nanometer to many micrometers. Additionally, we observed that there's a qualitative change in the confinement energy of DNA as we approach its persistence length. Now, these experiments are still ongoing, so what are some of the next steps that we're going to take? Well, we want to repeat these experiments using different length DNA. Additionally, we want to use lenses with different radiuses of curvature so we can examine steeper chambers or more shallow chambers. Finally, we want to turn our analysis to a counting-based analysis. The fluorescence analysis that we use today is subject to non-uniformity staining in the DNA. A counting analysis would be more rigorous. We want to then take these high-resolution films that we've seen, the different ones where the DNA is more stretched or more bundled, and compare this to polymer physics theories that have been applied in nanochannels, such as OJ and the GEN. And finally, although I showed you a system today using very simple fluorescently labeled DNA, we can then take this system and apply it to other ones, such as DNA-DNA interactions or DNA-protein interactions, and understand how these systems behave under extreme confinement. I'd like to say thank you to everyone at my lab, in particular to Professor Leslie, to Daniel Berard for his work on the click devices and helping him collect data, to Jason Leith, our postdoc, who helped set up our microscope and collect data as well, and of course to all our funding. And I really encourage you guys to listen to Daniel's talk later. He'll be talking more about our devices and their applications. It's quite interesting. Thank you.
or have you looked at the speed at which the DNA walks out from that confined region?
Yes, we do. So when we're studying the system, we're actually studying the equilibrium profile of the DNA. So after we form the chamber, we wait on the order of half an hour. It takes about half an hour for the DNA to reach its complete equilibrium, which is traversing several fields of view.
Any more questions from the floor?
All right, thank you very much, everyone.